Molecular basis of inheritance, part 3. In this video, you will be learning about DNA replication and the proof to prove that DNA replicated by semi-conservative process. RNA was the first genetic material and life evolved around RNA. RNA was a genetic material and also a catalyst, and hence it was highly reactive and unstable. DNA then evolved from RNA with modifications that made it more stable. DNA, being double-stranded with complementary base pairing, resists changes by evolving a process of repair. Watson and Crick proposed a scheme for replication of DNA. The scheme suggests that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental strand and the other strand that is newly synthesized. And this was termed as semi-conservative process of DNA replication. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together. That is A with T and G with C. Separation of the two single strands of DNA creates a Y-shaped structure called a replication fork. The two separated strands will act as templates for making new strands of DNA. One of the strands is oriented in the 3' 5' direction towards the replication fork, and this is the leading strand. The other strand is oriented in 5' 3' direction, that is away from the replication fork, and it is called the lagging strand. As a result of their different orientations, the two strands are replicated at different times. In the leading strand, a short piece of RNA called a primer, which is produced by an enzyme called primase, comes along and binds to the end of the leading strand. The primer acts as a starting point for DNA synthesis. DNA polymerase binds to the leading strand and walks along it, adding new complementary nucleotide bases, that is A, C, G, and T, to the strands of DNA in the 5' 3' direction. Here the replication is continuous. In the lagging strand, numerous RNA primers are made by the primase enzyme and binds at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in the 5' 3' direction. This type of replication is called discontinuous, and the Okazaki fragments are all joined up later. Once all the bases are matched up, an enzyme called exonuclease strips away the primer. The gaps where the primers were there are then filled by more complementary nucleotides. The new strand is proofread to make sure that there are no mistakes in the new DNA sequence. Finally, DNA ligase seals up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides are formed. Thus, DNA replication is called semi-conservative process. Following replication, the new DNA molecule automatically will wind up into a double helical structure. Watson and Crick suggested that DNA replication is semi-conservative and each daughter molecule contains an old and new strand. Matthew Meselson and Franklin Stahl provided a strong evidence for semi-conservative mode of DNA replication in Escherichia coli. Meselson and Stahl in 1958 conducted an experiment on E. coli which divides in 20 minutes to study the replication of DNA. In the experiment, they took heavy nitrogen, that is N15, and normal nitrogen with N14, the two isotopes of nitrogen, which can be distinguished based on their densities by centrifugation in cesium chloride. Meselson and Stahl cultured E. coli in a medium containing heavy nitrogen, that is in ammonium chloride, over many generations. As a result, N15 was integrated into the bacterial DNA. Later, they revised the N15 ammonium chloride medium to normal N14 ammonium chloride. At a regular interval of time, they took the sample and checked for the density of DNA. In sample number 1, after 20 minutes, the sample had bacterial DNA with an intermediate density. Sample number 2, that is after 40 minutes, contained DNA with both intermediate and light densities in the same proportion. Based on observations and experimental results, Meselson and Stahl concluded that DNA molecules can replicate semi-conservatively. Investigation of semi-conservative nature of replication of DNA or copying of the cells, DNA didn't end there. Followed by Meselson and Stahl's experiment, Taylor and colleagues conducted another experiment on Vicia faba, which again proved that replication in DNA was semi-conservative.